Hello and welcome, dear friends, to the final lecture of nitrogen cycle. Before venturing to the molecular biology aspects of this very important elemental cycle in the nature, I would like to motivate you towards higher understanding and higher learning by giving a small example of evolution. This example has nothing to do with nitrogen cycle, but dear students, I want you to emphasize how the higher knowledge or the higher quest for understanding the knowledge at the higher level is very important. You should have that quest in your mind. The evolutionary example which I would like to give you before starting the nitrogen cycle is that we as humans were on our four legs before we got upright and became human beings. We have a spine, we are vertebrates, chordates we say. So what made us come on two legs from four legs which we were before the actual evolution took place, actually before we became human beings? So that was our quest to see what is at the top. When we were on our four legs, we just wanted to see the sky. Maybe we wanted to see the moon, we wanted to see the stars. Maybe we then started jumping on our hind legs, two legs, and it took some millions of years for us to stand upright and see the sky and the stars. Not only that much, go on the moon, go on Mars, send space shuttles, fly in the air. All this development began with one single quest, one single urge to understand what is up there, what is up above the sky. So that urge first of all made a physiological change in us. We made our spine straight. All the animals you see, they are still on their four legs, except for some chimpanzees and monkeys. They are still trying. They are somewhere in the scale of evolution behind us. But that is that quest for knowledge, that quest for understanding the higher level of knowledge, which has made us human beings and developed our brains, which is connected to the spine, and it controls everything which makes us an intellectual. So why I am giving you this example is we come to a stage of nitrogen cycle where we are now trying to understand the deep science behind it, the knowledge behind it. Some of you who may find it difficult, some of you who may find it interesting, definitely you will not find it very easy. That is not a part of the curriculum. Easy or hard is not the issue. Lifting a 1,000 kilo stone may be hard, but understanding becomes easy when you try to explore it with what actually is happening at the molecular level, what is happening at the genetic level. So come, dear friends, we will see the nitrogen cycle as we should see from the level of a graduate, from the perspective of a graduate student who is going to become a postgraduate and a doctorate maybe. So till now what we have seen is just the basic aspects of nitrogen cycle. That is how the nitrogen moves around in the air, how it moves around just making transformations, undergoing transformation, making changes, drastic changes, subtle changes, due to which its form changes and it gets fixed somewhere and then again is released in the atmosphere. So this figure behind me sums it all, which we have seen in the last two lectures. The nitrogen cycle, how it cycles around the atmospheric nitrogen and from getting transformed into various forms back to nitrogen in the air. While going this, it undergoes a variety of physicochemical changes. One of the most important physicochemical changes is its oxidation state. If you see the transformations which are occurring with respect to nitrogen, we as microbiologists will be most importantly dealing with the microbial transformations. Of course, there are non-microbial transformations, there are symbiotic and non-symbiotic within the microbial transformations. That will be the point of discussion today, and how it is made possible at the genetic level or at the molecular level. So if you just see the genus, you can pause this slide whenever you think suitable and just go through the name of the organisms. These are the bacteria. Even archaebacteria are there which carry out the transformations of nitrogen. These are either free living, these are chemolithotropic, these are heterotrophic. I will categorize all this. So what actually these microorganisms do are they mineralize the nitrogen. What is nitrogen mineralization? It is conversion of organic nitrogen to more mobile inorganic state, which is known as nitrogen mineralization. As a consequence of this nitrogen mineralization, ammonium and nitrate are generated. We have seen that. And the organic nitrogen disappears. This takes place in two distinct microbiological steps, means these are carried out by microorganisms. And when we say these are carried out by microorganisms, you have to remember that there is certain level of enzyme production needed for this metabolism to occur. So this takes place in two distinct microbiological steps, ammonification and nitrification. Now what it actually happens during the course of reactions is that proteins and the waste products are microbiologically decomposed into amino acids, which are then ammonified, and the ammonia, which is formed by a certain group of microorganisms, that is Nitrosomonas species, they convert it into nitrite, and which by another species, Nitrobacter, is carried to convert it to nitrate, and this nitrate by yet another species of bacteria like Pseudomonas, they are fixing this nitrate and releasing back the denitrifiers, release it back to the atmosphere. Then again it is fixed in the form of ammonia, which we see as a cyclic process. So during this cycle, during this transformation, the nitrogen, it gets transformed into various molecular states, that is into different oxidation states, and these oxidation states vary from -3 of ammonia to +3 of nitric acid, +5 of nitric acid, +3 is of nitrous acid. So there is an increasing oxidation number. What is the oxidation number? It is the state of oxidation or the redox reactions between which the oxidation and the reduction reactions, which is called as redox reactions. The redox reactions in between which this nitrogen is shuttled. It is shuttled and gets converted by accepting or giving some electrons into various molecular forms. So that is oxidation state or the oxidation number. These are the oxidation number, -3 of ammonia to +5 of nitric acid. That is what the nitrogen gets transformed into. The total number of electrons which this nitrogen either gains or loses in order to form a chemical bond with another atom, like hydrogen, we have seen oxygen, so on and so forth. So the oxidation states of nitrogen in the valence shell, it has 5 electrons and it has 9 oxidation states, and it undergoes around 14 reactions, 14 redox reactions in the process, that is ammonia, nitrite, nitrate, so on and so forth. We have seen all these products in our previous lecture. So if you want a much more complex state of how these things are going on, this slide sums it all. You can pause this and just go through in detail. Of course, I will not spend much time on this slide because this is just to give you a fair idea of how the microbial transformation of nitrogenous compounds are carried out by enzymes which perform these 14 redox reactions. These redox reactions involve 8 to 9 key inorganic nitrogen species, 8 inorganic nitrogen types of different oxidation states. What is this oxidation state in a microbial transformation? It is an enzyme bound intermediate, and the redox states are shown here. That is, when all of you know what happens when a sugar is metabolized, it ends into a pyruvate. This pyruvate gets converted into acetyl CoA, It again gets associated with acetyl CoA. It enters into the TCA cycle, and there is one TCA cycle, EMP and TCA continuously going on, whether that is autotrophic bacteria, photoautotrophic bacteria, or any other type. There is a key cycle called as the EMP pathway. Linked to it is the TCA cycle, and the intermediates of the TCA cycle are the precursors for formation of amino acids. On one side of the cell there is a respiratory electron transport system, that is the RETC, which is continuously undergoing certain redox reactions where the terminal electron acceptor is oxygen, and it continuously provides NADPH2 where the electrons are shuttled and ATP is generated. This ATP is needed for nitrogen metabolism or the nitrogen cycle. So here you can see the ferredoxin, 8-ferredoxin, they are oxidized, 8-ferredoxin, it is reduced, oxidation reduction takes place, then dinitrogenase. In the nitrogenase enzyme complex of any microorganism, there are two important enzymes which are there, which we have seen, dinitrogenase reductase and dinitrogenase, and this collectively will be called as the nitrogenase enzyme system or the NIF genes, that is the product of NIF genes. I will come to that, but you just see how the redox reactions are shuttled. How these 14 redox reactions, they are, which involve 8 different types of nitrogenous compounds or transformed molecules. They are formed by redox reaction of ferredoxin, then dinitrogenase reductase, which is there is again a reduced form and oxidized form. It involves 16 ATP here, generates 8 electrons, and this again in its this dinitrogenase in its oxidized and reduced form gives rise to electrons, which is accepted by the reaction where ammonia and nitrates are formed. So this is the role of electrons in nitrogen cycle is very important, very vital, and the oxidation states of nitrogen, as you can see, the +5 of nitrate to -3 of ammonia. The nitrogen gas which is there, it is inert, 0, and nitrous oxide is +1, nitric oxide is +2, nitrate is +3, nitrite is +3, and nitrate is +5. Similarly, the hydroxyl amines -1, hydrazine -2, and ammonia -3. So from -3 of ammonia to +5 of nitrate, the oxidation states of nitrogen, it varies to a great extent. So this is very key in understanding the microbial transformations of the nitrogenous compounds. Now, having understood the role of electrons, if you see the types of nitrogen fixation, there are basically, it can be simply classified into non-biological type of nitrogen fixation and the biological type. Non-biological type, I have given you certain examples. So in the next slide I will give you some more just a recap of how non-biological takes place, but the main focus of discussion today will be the biological nitrogen fixation and trying to understand it at the genetic level or the molecular level. So nitrogen fixation can be biotic, that is biological, or abiotic, that is non-biological. And when it comes to non-biological, atmospheric nitrogen fixation like thunder, electric discharge, or the volcanic eruptions, etc. Even in industrial chemical fertilizer preparation, it is non-biotic process. In the biological process, symbiotic and non-symbiotic, these two are the process. In non-symbiotic, you can see aerobic bacteria like Azotobacter, which are freely present in the soil, anaerobic bacteria like Clostridium, which are also freely present in the soil, and blue-green algae like Nostac, etc., Spirula, Spirulina, Spirulinum, etc., which are present in moist environment. They are also, or lakes, sediments, etc., they are also fixing nitrogen. In the symbiotic type, you have those which have a symbiotic association with plants. So in the plants, we are always talking either about the roots or the stem. In the root nodule, they will form some nodules. They means bacteria will form nodules on the root or the stem. Like in root nodule formation, rhizobium and bradyrhizobium are the best examples, and stem nodulation is azorhizobium. As the mechanism of nodule formation remains same with root and stem with some minor differences. I will be mainly focusing on the biology of root nodule formation. Biology means the genetics of root nodule formation in this lecture. So we are discussing about the biological nitrogen fixation, but just a recap for you to have it in your subconscious how the non-biological nitrogen fixation takes place. Here the microorganisms are not taking any part. These are found in rainy season during lightning. Non-biological fixation either takes place in volcanic eruptions or found in rainy season during the lightning process, where these reactions they take place. We have seen all this reaction. Just one I would like to add here, that is the calcium nitrate formation, where huge calcium deposits are there. When acid rains fall, the acid soil has rich deposition of calcium. Nitric acid, when it falls through the form of acid rains in the soil, the calcium it combines with nitric acid to form calcium nitrates. These are also used by certain organisms and the nitrogen gas may be released. This is one part of the non-biological nitrogen fixation. But our main discussion here today is biological nitrogen fixation. These microorganisms which are capable of fixing the nitrogen, those are the key players in the biological nitrogen fixing process. Why biological? Because microorganisms are involved and either freely they fix nitrogen or they associate with the plants and then they fix nitrogen. So industrial nitrogen fixation and biological nitrogen fixation, if you see here in this slide, let me zoom if I can. So if you see here, the process, yes, here if you see the biological nitrogen fixation, the non-diazotrophs, they are the industrial nitrogen fixation, and diazotrophs, that is the bacteria, they are called as diazotrophs, and they fix the nitrogen directly from the air. Of course, there are certain bacteria which can associate with the plants and they form the process of nitrogen fixation by symbiotic method. So the nitrogen cycle in the biosphere is carried out by diazotrophs, that is the bacteria which are responsible for fixing the nitrogen. So biologically speaking, you can categorize into two types: agricultural systems and natural systems. The natural systems, which are free living bacteria and the plant associated bacteria, there is a jargon. Jargon means the language which is used in sciences. They use the terminology PALN, plant associated nitrogen fixation, PANF, and biological nitrogen fixation, BNF, plant associated nitrogen fixation, PANF, free living nitrogen fixation, FLNF. So plant associated nitrogen fixation and free living nitrogen fixation, these are the two types of nitrogen fixation when you talk about agricultural and natural systems. In natural systems, when it is not controlled by man, when it is freely going out in the nature, the plant associated legume rhizobia are there, non-legume plants are there, then free living cyanobacteria, heterotrophic bacteria, autotrophic bacteria, these are there. In the agricultural system, man controlled crops are there, and free pastures and fodder, means in the freely growing crops which are not cultivated by humans, but still the free pastures which are there, they are also associated with plants, that is the nitrogen fixing by microorganisms can be plant associated or free living. So this is all about the biological nitrogen fixing. In this, if you see these diazotrophs, how do you define the diazotrophs? Just we have seen diazotrophs are bacteria as well as some archaea. Bacteria as well as archaea which fix the atmospheric nitrogen gas into a more usable form, such as ammonia. A diazotroph is a microorganism, you can say, which has the ability to grow without the external nitrogen source. So when we are growing these diazotrophs in the laboratory, we use a media which is nitrogen free. So mannitol is the carbon source for such free living bacteria like Azotobacter, and we have a nitrogen free mannitol broth, a broth which will not contain nitrogen source. Why? Because the air which is present in the headspace of that flask, When you prepare a media which is not containing even a single mole of nitrogen, the nitrogen is provided by the air present in the headspace of that flask, as well as above the cotton, beyond the cotton, which is present in the atmosphere. Through the cotton, it has a high respiratory rate where it can breathe in and out the oxygen and the air for utilization of nitrogen. So these are diazotrophs, which are bacteria as well as archaea, which can fix the atmospheric nitrogen into a more utilizable form such as ammonia, and these are those microorganisms which are having the ability to grow without the nitrogen source present in the media. They utilize the external source of nitrogen from air. Okay, it need not be added in the media. Some of the best examples are Azotobacter, Rhizobia, Frankia, Azospirillum, etc. So these diazotrophs can be heterotrophs or photoautotrophs. As I just said, Azotobacter, it is a heterotroph, whereas the Rhizobia and Frankia and Azorhizospium, these are the photoautotrophs. Heterotrophs, these can be free living and symbiotic. Just like the photoautotrophs, they can also be free living and symbiotic. These heterotrophs can be aerobic, facultative, anaerobic or anaerobic, whereas these photoautotrophs, they can be cyanobacteria, some other bacteria, or when they are free living and when they are associated with the plants, they can be nodulating bacteria or non-nodule forming bacteria. So you can find the examples here of each of these species. Again, I recommend you to pause this video here and just take into the examples, give some time, see this slide in high resolution. You can change the resolution if this screen is blurry. You change the resolution to 720 or 480p, and you can see the names here in aerobic: Azotobacter, Azomonas, Bigerinkia, Rhizobium, Azospirillum, etc. So these are the microorganisms which are categorized according to their nitrogen fixing capabilities. Now, nitrogen fixation by the microorganisms, we have already seen that the bacteria which are capable of fixing the nitrogen, they are called as diazotrophs, and they have the capacity to fix by the help of an enzyme called as nitrogenase. My next part of the discussion will mainly focus on this enzyme, this wonder molecule called as nitrogenase enzyme. This enzyme has the capacity to break this bond which is broken by lightning. It has the capacity to break the bonds among the molecular nitrogen and combine it with hydrogen. Okay, nitrogenase only functions when there is no oxygen, means it is very sensitive to oxygen. There is complete removal of oxygen, at least at that point where nitrogenase is functioning, is needed. And this is accomplished by many means. We will see how it happens in cyanobacteria, how it happens in Azotobacter, and how it happens in root nodulation bacteria, that is Rhizobium. So these bacteria, usually like cyanobacteria, they form heterocyst. I will come to this heterocyst in my next slides again. But here just understand, nitrogenase enzyme is the key enzyme which is involved in breaking of the bond and adding a hydrogen by which the nitrogen gets fixed. Now this slide may feel very complicated, but just neglect all this matter which is written on this at this level of graduation. If you are interested, you can read all the functions of each gene which is responsible for fixing nitrogen. This is the genetic map of nitrogen fixation. You can see NIF genes at the two ends, and in the middle there is nodulation formation genes given here. So this nodulation or the nod genes, nod M, nod L, nod E, nod F, nod D, A, B, C, D, E, etc. These genes are involved in nodulin protein formation. Nodulin is a protein which is responsible for inducing a root nodule in leguminous plants. I will explain that. But here what the main aim of this slide is, just as I said, forget, there are 1, 2, 3, 4, some 10 to 15 genes here. These gene set is called as NIF genes. So you can see here 1, cluster of NIF genes is there, and at the another end there is a cluster of NIF gene. This is a DNA of some microorganism, any microorganism. Actually, the best studied microorganism for studying the NIF genes is Klebsiella pneumoniae. This Klebsiella pneumoniae is capable of fixing nitrogen, and its nitrogen fixing genes have been completely mapped. It is a NIF operon. Operon is a cluster of genes. Many genes are there which collectively are responsible for regulating, modulating and fixing the atmospheric nitrogen by forming proteins which control this very complex and beautiful process of nitrogen fixation. So many genes have been identified, their roles have been identified, as you can see here in this slide. As I say, don't go, it is not necessary that even you memorize these genes. It is just this slide is just to show you the complexity or the beauty of nitrogen fixation by microorganisms. So many genes, so many collective effort which is there, and they do which lightning does in a second. They fix the atmospheric nitrogen either in association with plants or free living, just as it is in the soil. And if you go inside the microorganism, see its DNA structure, see its genes. What is a gene? Gene is a functional unit of a DNA. What is the function? The function is protein synthesis. Function of a gene is metabolism, to felicitate metabolism. And this metabolic activity here which we are focusing on is nitrogen fixation. So nitrogen fixation is a activity which is carried out by genes. So one metabolic reaction, there is one gene. So if you think nitrogen is fixed in one step, there will be one gene, but it is not as simple as that. Nitrogen is fixed, it is cycled, it is circulated, and it is transformed into various forms by a variety of reactions, and each reaction is needing an enzyme. And this enzyme, collectively all these enzymes are called as nitrogenase enzyme system. And the collective name of all the genes responsible for all these enzymes, I will call it as proteins. All these proteins is called as NIF gene. So the genetics of nitrogen fixation involves NIF gene, which is responsible for nitrogenase enzyme. So nitrogenase is not a single enzyme. Remember that. Okay, so this is what this slide intends to tell you. What are NIF genes? NIF genes, there is a trend to say NIF gene also, but when you say NIF gene, it is somewhat paradoxical. It is very strange to call it as a NIF gene, because it is a cluster of genes which is responsible for coding of proteins. Again, I am not using the word enzyme here. Why? Because all enzymes are proteins, but not all proteins are enzymes. But whatever a gene will be producing, that has to be a protein. Protein is produced first. That protein can be changed, transformed by some active site modification into an enzyme. So we will use in a broader sense the term product of a gene as a protein. So the NIF gene is a gene responsible for coding of different proteins which are associated with the fixation of atmospheric nitrogen. These NIF genes are found in nitrogen fixing bacteria, all heterotrophs, all diazotrophs, as well as cyanobacteria. So if you see each point, you will understand the significance of this NIF gene. First, the NIF genes, remember, are involved in fixing the nitrogen, atmospheric nitrogen. NIF genes are not single genes, but they are a part of a NIF gene, NIF operon, I can say, which is a cluster of genes, and they are found in nitrogen fixing bacteria. These genes occur as an operon. What is an operon? It is an operational system, a collectively operational system which regulates the fixing of nitrogen. They occur as an operon in the free living anaerobic nitrogen fixing bacteria like Klebsiella pneumoniae, the most studied nitrogen fixing bacteria, that is Klebsiella pneumoniae, Rhodospirillum rubrum, and Rhodobacter capsulatus, to name a few. There are many other organisms also. These genes may also be found on plasmids. Means when you talk about a genetic system of a cell, the main genetic system is the DNA and the secondary genetic system is the plasmid, an extra chromosomal entity, which may have certain genes which are present on the chromosome or not. they may contain exclusively certain genes. So this NIF gene can be present on the DNA or it can also be present on the plasmid. That has to be remembered. And together with other genes like NOD genes. What is NOD genes? NOD genes is the gene which induces nodulation, root nodulation. I will explain that. So in symbiotic bacteria, this is there, the NOD genes. Like in Rhizobium, you have a root nodule. So NIF gene along with a NOD gene. NIF gene is involved for fixing nitrogen and NOD gene is responsible for creating a root nodule. Root nodule produced by the induction of NOD genes is responsible for giving a safe house, a shelter, a niche for the symbiotic bacteria. These NIF genes, remember, the next point is codes for proteins essential in nitrogen fixation such as nitrogenase enzyme, and some other regulatory proteins may also be produced. Nitrogen fixing is very important, as we have known, because most of the living organisms are unable to metabolize or directly use the atmospheric nitrogen, and they require it in some utilizable form. So these bacteria, they are the key players who make us avail or they make us available the utilizable form of nitrogen. For humans, we eat plant and plant products, and that is the source of nitrogen. So that is NIF genes for you. Now with respect to nitrogenase enzyme system and the NIF genes, let us try to understand the metabolism or the biochemistry of nitrogen fixation at the molecular level. The basic requirements for nitrogen fixation is nitrogenase enzyme complex and a hydrogenase enzyme complex. Hydrogenase is also needed because atmospheric nitrogen has to be, the three bonds have to be broken and a hydrogen has to be added there. So that will require hydrogenase. And one more important thing is that as nitrogenase enzyme, remember this is a very important point, as nitrogenase enzyme is very sensitive to oxygen, there should be some mechanism by which the nitrogenase enzyme should be protected from oxygen. So this is the requirement. Then ferredoxin proteins, there is a molybdenum also required, I will show you next. Hydrogen releasing system or the electron donors like pyruvic acid or glucose, sucrose, that is the TCA cycle, and its electron transport system is needed. Why TCA cycle? Because sugars, when they are metabolized, it gives rise to some intermediates which forms amines, amino acids, and that is utilized here. That and the ATP which is generated in the RATC, that is utilized here in the nitrogen fixation process by the nitrogenase enzyme. There is a constant supply of ATP needed, and coenzymes and cofactors like TPP, acetyl-CoA, inorganic phosphates, magnesium ions are needed. Carbon is needed. A carbon is to nitrogen ratio is always fixed when there is a nitrogen cycle or a carbon cycle. That is totally another area of study, that is the carbon is to nitrogen ratio, which is very important in the carbon cycle as well as the nitrogen cycle. These both cycles, they are interrelated to each other. You cannot separate it from one another. There is a ratio, and that ratio is determinantal in the in how much amount of carbon or nitrogen is fixed, and that accounts for the global cycles. So apart from that, cobalt and molybdenum are required, and carbon compound, as I said, for the carbohydrates for metabolism is needed. Now if you see the nitrogenase enzyme, which plays a very key role, is very active in the anaerobic condition. I am saying it is active in anaerobic condition does not mean that the bacteria, they are anaerobic. No, the bacteria, they are highly aerobic. The nitrogenase enzyme system needs to be worked out in an anaerobic condition. And how that is achieved? That is achieved by a high respiratory rate. I will explain that. So nitrogenase enzyme, which plays a very key role in the nitrogen fixation, requires anaerobic condition. The bacteria, they themselves are not anaerobic, but the nitrogenase enzyme system needs an anaerobic condition. This nitrogenase enzyme is made up of it is a metalloprotein which contains iron and molybdenum. So it is made up of two metalloprotein subunits, non-heme, it is called as a heme and a non-heme. non-heme iron, that is ferrous iron protein or dinitrogen reductase, ferrous iron protein or dinitrogen reductase, and iron molybdenum protein, this is iron and molybdenum protein or MOFE protein, which is again a dinitrogenase enzyme complex. So this iron reacts with ATP in the reaction and it gets reduced. The second subunit, which is also reduced, and the net reaction is the atmospheric nitrogen getting converted into ammonia. As you can see here, atmospheric nitrogen getting converted into ammonia. So this is basically the core of a nitrogenase enzyme. The reduction of atmospheric nitrogen to ammonia, it requires 6 protons and 6 electrons. 12 molecules of ATP are required. This is the bioenergetics of the nitrogen fixation in the biological system by microorganisms. So the reduction of atmospheric nitrogen to ammonia, it requires 6 protons, 6 electrons, 12 molecules of ATP are required, 1 pair of electron, it requires 4 ATP, and the reaction you can see here, ammonia is formed. Hydrogen produced here, it is catalyzed by the enzyme hydrogenase, and protons and electrons are formed. These protons and electrons are the ones which are responsible for transforming the oxidation states of nitrogen in various forms. So if you see the pathway of nitrogen fixation in the symbiotic type of biological system, where the bacteria, they get associated with the root nodules. So here you can see 6-glucose phosphate, it acts as electron donor. Sucrose, it is synthesized in leaves. In cyanobacteria, in the photosynthetic apparatus, it takes place. Sucrose in the roots, glucose and fructose converted into glucose 6-phosphate enters into the EMP pathway. 6-phosphate is converted to phosphogluconic acid. Glucose 6-phosphate then is converted into so on and so forth, and it enters into the TCA cycle. In the another reaction where NADPH, it donates electrons to ferredoxin, protons released from the ferredoxins, they are reduced, and a reduced ferredoxin, it acts as an electron carrier. This donates the electrons to Fe protein of the nitrogenase enzyme system and it gets reduced. The iron part of the nitrogenase enzyme system, it accepts ATP, that is the reduced iron protein complex of the nitrogenase system, it combines with ATP, magnesium ions are required here and it gets reduced. The second subunit is activated now, it donates electrons to atmospheric ammonia and it converts atmospheric ammonia to nitrogen. The enzyme is removed free. Once the ammonia is formed from the reaction, the enzyme is left free and it is ready for the next reaction. So you can see here the molybdenum enzyme complex combines ammonia, and once ammonia is formed, that is, it is the enzyme complex is set free from the ammonia and ammonia is released in the soil. So this symbiotic nitrogen fixation, that was all about the biochemistry of the nitrogenase enzyme system. Now let us see some examples of the symbiotic nitrogen fixation and non-symbiotic nitrogen fixation with respect to microorganisms. Okay, you should remember that we are exploring the key players of the nitrogen cycle here. We are trying to understand their molecular biology, what happens in the biological organelles of the microorganism as well as the plants if it is a symbiotic nitrogen fixing process, like in the rhizome legume association, rhizobium and the legume association. This is a leguminous plant. Rhizobium forms a root nodule, rhizobium legume association, and which forms root nodules. It is also seen in soya bean. It is also seen in pea plant. Okay, so these are called as the rhizobium leguminosarum root nodules. It has a pink coloration. If you see this, I will zoom in again. Let us see if you can find, if you can see this. Yes, here. So this red coloration which is there, this is a close-up of a root nodule of rhizobium, this pink color owes to the name leg hemoglobin. It has nothing to do with hemoglobin. Just if you see it deeply, you will see it forms a red color. Maybe that is due to iron which is there. So fixing pink color is termed as leg hemoglobin, which is a protein which carries the oxygen in the bacteria. This you can see here the picture of root nodules of soybean by Bradyrhizobium japonicum, and this is the pea plant. Okay, so the symbiotic nitrogen fixation, the main aim of this slide is to tell you that by associating with certain plant families of the leguminous plants, primarily the legume forming species, the bacteria, they form the largest single biologically nitrogen fixing species in the biosphere. If you see the enzymology of nitrogen fixation in rhizobium, what are the steps, how the stages are formed, and how the root nodulation takes place. This occurs in prokaryotes, that is the association of rhizobium with plants. Rhizobia, they fix nitrogen in symbiotic association with the leguminous plants, first thing. Then they fix nitrogen for the plant with only those plants which can provide it carbohydrates for growth and a shelter. It is a symbiotic association. Both of them are benefiting. How? The bacteria, it is getting carbon source and protection, whereas the plant, it is getting nitrogen fixed for its nitrogen requirements. All nitrogen fixing systems appear to be identical. They require nitrogenase enzyme which reduces the ferredoxin in the nitrogenase enzyme complex, as we have seen, combines with ATP, and all this is a part of expression of the NIF genes. Why does the nitrogenase require so huge amount of ATP? Because atmospheric nitrogen reduction to ammonia is thermodynamically favorable, means it involves thermodynamics. Heat is evolved in it. It is transformed from one form to another form. However, the activation barrier for breaking the three bonds, triple bond of nitrogen is the huge energy is required. 16 ATPs are needed, and those are provided by the RATC of the bacteria, and this is how the nitrogenase enzyme system works and is get activated. Now if you see the detailed biology of nodulation or the nodule process, it involves nodulin proteins. Nodulin proteins are secreted by bacteria. That is, the bacteria themselves, they have certain genes called as the nod proteins. Along with the NIF genes, they have nod genes which secrete nod proteins. And if you see the root hairs of rhizobium, these symbiotic bacteria, first they recognize and attach to the root hairs. Ricardicin is the protein which is secreted, and ricardicin is it is not an enzyme, a protein which mediates the attachment of these symbiotic bacteria to the root hairs. Once the rhizobial cell, they attach to the root hairs, they excrete the nod factors, that is the nodulin proteins, and this causes the root hairs to curl. The root hairs, they curl inside. Curling of means inside, they form a curve, and invasion of this rhizobium takes place by they getting penetrated inside the root hair, and once they get inside, they start multiplying, means it is a sort of infection. This is an infection, infection thread is followed. Bacteria, you can see now, once the curling takes place, you can see the fourth figure here, the bacteria in the infection thread, they grow towards the root cell. Inside towards the root cell they grow, and the invaded plant cells, they are stimulated to divide. Once they start dividing, you can see here in the last picture, they form nodules. Okay, formation of a bacterioid state means it is just like a colony formation on the agar. They form a root nodule inside the root. This is all carried out by virtue of the nod factors. Bacteria, they are attracted to the root. they attach themselves to the root and secrete specific. What are these nod factors? They are oligosaccharides and signal molecules. The signal which initiate the first stage of the nodulation. So this whole process can be summarized in 7 steps. That is the chemical recognition of the root by the rhizobium. Chemical recognition, what is the chemical? Ricadacin is the chemical which is involved in recognition. Then second is the curling of the root hair. Third is formation of the infection threads, that is long infection threads inside. Because of the polysaccharide, it forms a thread-like structure. Then invasion of the roots by the rhizobia. Fifth is nodule tissue formation, that is nodulation starts at a certain level. Bacteria, they convert the bacterioids and begin to form the nitrogenase enzyme system inside this. They activate the nitrogenase enzyme, and the legume, that is these leguminous plants, which is legume means the root nodules, this provides the rhizobia with carbon, means the root nodule roots. They supply carbon source to the bacteria which are growing inside these nodules, and in turn the rhizobia, it fixes the nitrogen for the plants. So this is the root nodulation process in symbiotic type of bacteria. Nodulins, these are the proteins, and as I have shown you earlier in this genetic map, in the cluster of NIF genes, if you see the NIF genes at the terminal end, there is a complete set of nod genes. These nod genes are responsible for producing nodulins. Nodulins there are of 3 classes of nodulins: early nodulins, late nodulins, and the nodulins which remain permanent inside the system. So these are involved in bacterial attachment, root hair invasion, bacterial development, nitrogen fixation, and nodule senescence, that is the signaling of the root nodule. So again the same picture, as you can see, summarized in this. This is the actual picture, this is schematic, and you can see here the root hair, they are attracting the rhizobia, then infection, then the thread formation, curling takes place, and the root nodule develops here. Okay, so this is the genetics of nitrogen fixation. Now root nodule formation in the leguminous plant has some interesting facts. 2500 species of family Leguminosae, okay, produce root nodules with rhizobium species. Means this rhizobium bacteria can associate with 2050 to 2500 species of plants, different species of. So you see the biodiversity it has achieved by infecting and forming a symbiotic association. They fix nitrogen inside the root nodules of these thousands of different plants. The association, it provides food and shelter to the bacteria, carbon source, and protection from invasion of other like bacteria eating higher cells which are there. Okay, so this protection is there, carbon is supplied, and nodules, they may be buried in the soil even after harvesting, and they still continue the nitrogen fixation. That is the signaling which I had mentioned. Non-nodulating types are also there, lichens, cyanobacteria, nostoc, etc. This is about the symbiotic type. Now let us see the non-symbiotic type of bacteria. In non-symbiotic, there are 2 examples. One is cyanobacteria, the other is azotobacter. So in cyanobacteria, the nitrogen fixation takes place with the help of heterocysts. So what are these heterocysts? These heterocysts are specialized organelles, or as you can see here in filamentous cyanobacteria, this is one differentiated cell, a cell which is somewhat different from the regular photosynthetic cells, which is somewhat swollen, and it has a thickened cell wall. This is the schematic diagram, you can see it is a thickened cell wall, and this protects the nitrogenase enzyme system. This is a nitrogenase enzyme which is obtaining the nitrogen from, that is the photosynthetic cells. As you can see, they are fixing the atmospheric nitrogen and bringing it into the cells, but this nitrogen has to be converted to ammonia with the help of nitrogenase enzyme, and this nitrogenase enzyme has to be protected from oxygen. So to provide it a micro anaerobic environment, These heterocysts are responsible. Heterocysts, are also called as heterocytes, are specialized nitrogen fixing cells which are formed within the regular cells. They are differentiated by their swollen nature, and they are having this structure to provide a micro anaerobic environment to the nitrogenase enzyme system. So you can see the photosynthetic cells, they are fixing or forming the succinate, malate, fumarate via the TCA cycle, whereas the leg hemoglobin is accepting the oxygen here and not allowing the oxygen to interact with the nitrogen fixing process mediated by nitrogenase enzyme. The ATP is provided by the electron transport system. Here huge amount of ATP is required for this reaction, and atmospheric nitrogen which is fixed by the adjacent cells in the cyanobacteria. This adjacent cell which is fixing the nitrogen, with the help of the nitrogenase enzyme converting this atmospheric nitrogen to ammonia, requires an anaerobic environment. We will call it as a micro anaerobic environment, and that is provided by the heterocyst, a thickened cell wall structure. These are formed during nitrogen starvation by some bacteria like the cyanobacteria, nostoc, anabena, etc. There is a sort of cyst formation in Azotobacter also, but its structure is somewhat different. So here you can see these are specialized organs which are helping the nitrogen fixation process. The nitrogen fixation in Azotobacter is also very unique in nature. These are the free living type of bacteria, and this forms the last part of our discussion. As an example of free living nitrogen fixing in soil, I would like to quote the uniqueness of Azotobacter here. These are the plants, and these soil, in the soil away from the root, as you can see, nitrate and nitrogenous organic compounds, these are exported into the root from a bacterial activity which is far away from the root. So there are Azotobacter nitrifying bacteria are there. You can see three different colored coding is done to show that three different process by three different types of bacteria are going out in the soil. Nitrogen fixing bacteria, this is the Azotobacter which I am speaking about. There is ammonifying bacteria, there is nitrifying bacteria, and there are denitrifying bacteria. So nitrogen fixing bacteria, these fix the atmospheric nitrogen away from the root system, away from anything, and they fix the nitrogen, converted into ammonia in the soil. From the soil, these nitrifying bacteria, they act on the ammonia and convert it into nitrate. Nitrate is then again uptaken and it is exported into the plant system in the form of nitrogenous compounds. So this genus Azotobacter, this is a very unique type of bacteria. It is mostly studied in the industry for not only its nitrogen fixing capabilities, but also the polysaccharide gum which is formed. It forms biogum, and these biogums are very popular nowadays in producing biodegradable plastics. Azotobacter also has one unique feature of forming huge amounts of reserve food material. That is a totally different aspect of study, but this reserve food material, that is the poly beta hydroxybutyrate granules, which are formed inside the Azotobacter in certain deficient conditions, they are also used as the bioplastic production. So that is industrial significance of Azotobacter. But here my main aim of explaining Azotobacter as a free living nitrogen fixer is with respect to the NIF genes and the nitrogenase enzyme system. Again, the genus Azotobacter, it comprises of huge species of bacteria. A wide variety of species are present which are categorized in the Burgess Manual of Systematic Bacteriology. So it was discovered in 1901 by Dutch microbiologist and a botanist Martinus Beijerinck. So Azotobacter beijerinckii species is also there, which is a nitrogen fixing bacteria named after the Dutch scientist who discovered Azotobacter species. So Martinus Beijerinck in 1901, he discovered this Azotobacter, and he is one of the pioneers in environmental microbiology. Beijerinckii, he selected and described the species Azotobacter crucocum, which is the first aerobic free living nitrogen fixing bacteria. Azotobacter crucocum, they are characteristic of brown pigmentation. They can be easily grown in your laboratory by using a nitrogen free mannitol broth initially and then transferring it into a nitrogen free mannitol agar. So these Azotobacter species, they are, as I said, free living, they fix nitrogen in contrast to the Rhizobium species which are associated with the plants. Now the nitrogen fixing without any symbiotic relationship with the plant itself is very important in the global nitrogen biogeochemical cycle. Nitrogen fixation here also in Azotobacter is inhibited by availability of nitrogen source. If nitrogen is added in the media or if nitrogen sources is present in and around the media where it is growing, it will not fix nitrogen. It fixes nitrogen only in conditions where there is nitrogen depletion, nitrogen deficient conditions, not sufficient conditions. Okay, so nitrogen fixation by Azotobacter is inhibited in the presence of nitrogen. Azotobacter species, they have a huge range of enzymes which are needed to perform the nitrogen fixation. Ferredoxin is one of the component which is involved. Hydrogenase enzyme is required and nitrogenase enzyme system is also required. It has a molybdenum enzyme system. The nitrogenase, molybdenum, ferredoxin which we have seen is involved in it, and the process of nitrogen fixation by Azotobacter, it requires great amount of ATP. So nitrogen fixation is a highly sensitive process, just like we saw in the symbiotic type. It is very sensitive and oxygen sensitive to oxygen. So this Azotobacter, they have developed a process of rapid or very intense respiration rate, a high respiration rate, a very significant and intensified metabolism which reduces the concentration of oxygen in the cells. Means the respiratory rate of Azotobacter is so high that the oxygen, it never reaches the nitrogenase enzyme system. A high respiratory rate and a rapid consumption of oxygen at the surface of these diazotrophic bacteria, it prevents the oxygen from reaching the nitrogenase enzyme, and thus it is not inactivated. Also, along with this, there is FeS2 protein. This is a nitrogenase protective protein called as FeS2. This is usually seen in Azotobacter vanillandi and some of the Azotobacter species. Some research is going on in this, but there is a special nitrogenase protective protein along with the high respiratory rate. The question is, how does Azotobacter protect its nitrogenase from the oxygen? The answer is, it protects by involving itself into a high respiration plus the FeS2 protein and protects the nitrogenase from the oxygen. Otherwise, nitrogenase is sensitive to oxygen. Azotobacter species have several types of nitrogenase. Most common is the molybdenum iron containing nitrogenase, which I have explained earlier. There is an alternative type which contains vanadium also. Some species of Azotobacter, instead of molybdenum, they contain vanadium, and it is independent of molybdenum ions, and it is more active than the molybdenum ferrous nitrogenase enzyme, and this vanadium containing nitrogenase is very active in psychrophiles, that is low temperature Azotobacter species. Azotobacter species growing in low temperatures are usually having vanadium in their nitrogenase enzyme complex. So there is a new area for research, that is low temperature nitrogen fixation. Azotobacter species, some of the species can fix nitrogen at low temperatures as low as 5 degrees centigrade, and at this low temperature the nitrogen fixing capacity is 10 times higher than the normal temperatures. So even in Azotobacter, all these nitrogenase enzyme is controlled by NIF genes. So this is all about the nitrogenase enzyme system, the bacteria involved in it. Symbiotic type we have seen, the non-symbiotic free living we have seen. This is electron microscopic picture of Azotobacter species. This is Azotobacter. These are rod shaped bacteria, and this is some 20,000 times magnification in electron microscope on a grass, switchgrass root. On a grass root it is growing, and you can see the rod shaped bacteria, and it produces a slimy layer along with it. So this is all about nitrogen fixation. So nitrogen fixation is just a part of the global cycles, which involves carbon cycle, nitrogen cycle, phosphorus cycle, and the sulfur cycle. So in our next lecture we will be dealing with the phosphorus cycle. So I hope you understood all the facts and the interesting points behind the nitrogen cycle. We started with a very simple explanation of nitrogen cycle, and we explored to the molecular level, to the genetic level of this nitrogen cycle, and I hope the quest for understanding the higher level should always remain, and it should take you, that should be the quest which will take you to the PG level or the postgraduate level, only after which you will become masters in your subject. So the very best of you all to go through the lectures once again and have a aim of becoming the masters in the subjects which you desire. So till then, goodbye, happy learning.